All right, good morning and welcome back. Today is Thursday. We are already on the third day of the workshop. I hope you have so far gaining some knowledge, some experiences, and also a lot of interactions. And so the next 1 hour, I'm going to talk about molecular mechanisms in hypertrophic cardiomyopathy. So it's kind of how genetic variants can lead to develop the disease. Again, we talked about yesterday and day before yesterday, the diagnosis, the variability, the onset, a lot of clinical aspect. But today we are going to focus on how genetic mutation in sarcomeric genes resulting in hypertrophic cardiomyopathy. Again, in a research lab, how do we study that? What are the tools we are going to use? What are the models we are going to what are the models we are going to use in the lab and then study? So it's kind of as a scientist aspect, how do we do all the experiments in the lab to test hypothesis? Again, being a basic scientist, I also do clinical study, going to the community, interacting with people, and then do my research. So it's kind of, I'm going to cover a lot of aspect. So again, we went through this hypertrophic cardiomyopathy. What does it mean? The phenotype, and then what are the diagnoses? The thing is that when you or someone remember hypertrophic cardiomyopathy, it's a #1 cause of sudden cardiac death. That is always been reported everywhere, and people in young athletes specifically, they die if they do very severe exercise. So again, we talked about all the variabilities in the disease development, starting from the cardiac structure, the thickness, chamber size, etc., and then the penetrance. It's kind of what time in your life the disease can develop or show the symptoms, and then cardiac arrhythmias, histology, all this aspect. Then also we covered about the modifying genes, secondary risk factors, and then lifestyle choices. So these are again, we don't know how these are interacting to the disease development, and again, lot of things to be learned. So the genetic of hypertrophic cardiomyopathy, again, it's a pure genetic disease. Predominantly mutations are in the sarcomere gene, mainly involved in muscle formation, muscle structure, muscle function. So we are in the next 10 minutes, we are going to look what are these proteins and how they work in the cardiac muscle. So this one is a real, numbers, like how our cardiac system in a day-to-day activities, it's almost 72 times per minute, 100,000 times per day. That is a lot. So it's more than an engine non-stop from day one of life formation, and then before you die, this is the last organ to be failed. Again, pumping 5 to 7 liters blood in 1 minute, and then 7,600 liters per day. That's a lot. So just assume, this is happening in your body, in your system, every time, every minute, every second. So you can't just stop this one. So it's a very important organ. Again, you can see the coordination. Blood come into the right ventricle, and then go back, and then pure get oxygenation, left ventricle, and then go out. So it's kind of constant process, but again, a lot of other organs also get involved in the pumping process. If you're really going back to the history of cardiac system, we always, in the ancient time, heart is always referred where we have our life. It's also been referred to character. For example, this person is a very good heart person. Somehow they related the character and the emotion to the heart. And then people believe that all our life, soul, are into the heart. So that's another belief. And then it's kind of symbolic of, religion. This is in 1530. It's a Lutheranism, a symbol. They introduced the heart. And then in the 17th century, it's become a love, symbol of love. So again, you can see how heart can be related to our emotion, feelings, and also character. So it's a very important organ to study anyway. This is the video kind of showing you, look at this one, where we have the heart attack. So heart attack is different from sudden cardiac death. Sudden cardiac death comes through cardiac arrhythmias and hypertrophic cardiomyopathy, but this is a myocardial infarction where there is a blockage in the artery, so such a resulting lack of blood supply to the region, and so the region can die. As a result, heart can stop working. So this is called a myocardial infarction or heart attack. It's completely different than sudden cardiac arrest. Sudden cardiac arrest, people passed within a second, just stopped. Whereas myocardial infarction, they get all the pain, they feel it, they know that, but you need to go to the hospital to be treated to remove the blockage. So that's kind of the major difference. So here again, I don't know whether you can see all these letters from the heart all the way to the muscle, and then we have a single cardiomyocyte. So cardiomyocyte is the cell, single cell of the cardiac muscle. It's only in the heart, not anywhere else. Skeletal muscle, they have different cell types, but both skeletal and cardiac comes under the muscle type called striated muscles, whereas smooth muscles are different because they don't have structures like defined structures. So the cardiomyocytes, and then within the cardiomyocytes we have the myofibrils, and then you look at the myofibrils, we have the sarcomere. Sarcomere is the unit of muscle contraction. So here this is a one sarcomere from Z line to Z line, and then in the middle we have the M line. So in this sarcomere is the functional unit of the muscle. So within the sarcomere we have all the proteins like thick filament proteins, thin filament proteins, and etc., which we are going to look into it now. So if you do electron microscopic analysis of the heart, what you see is predominantly the sarcomere or the cardiomyocytes. Within cardiomyocytes you have the sarcomere, which is surrounded by mitochondria. So again, mitochondria gives the energy for the muscle function, whereas the sarcomere generate the forces. So again, you can see this is the Z line to Z line. It's called one sarcomere. In the middle we have the M line. And the dark line, they are the thick filaments. In between two thick filaments, we have the thin filament proteins. Again, within the heart, 75% of the cell types are cardiomyocytes. But we also have endothelial cell, fibroblast, all other cells within the myocardium. So again, this is the schematic diagram of the sarcomere. You have thick filament, titin, and then myosin heavy chain, and then the myosin binding protein C, and then thin filament proteins, the troponin complex, actin, alpha tropomyosin. So these are the major sarcomeric proteins, and then the myosin heavy chain has two light chains, regulatory light chain and essential light chain, and those are attached to myosin heavy chain. Again, myosin is the motor protein. We talked about that yesterday. So again, if you look at how this contractile property occurs, a lot of signaling events can alter the force, the speed, all the property of contractility can be modified through so many factors which are listed here. For example, beta adrenergic stimulation, such as activation of catecholamine. You can see all these pathways can get activated, the kinases can be activated, resulting that they can directly come to the sarcomere and then activate or inactivate the function of the muscle. So today companies are focusing on, instead of going through all the pathways, can we directly target sarcomeric proteins and then treat the disease hypertrophic cardiomyopathy. So again, sarcomere is the contractile unit. We have thick and thin filament proteins. So you can see how they are. So thin filament proteins, primarily actin, and then we have the troponin complex, and then alpha tropomyosin. So within troponin complex, we have three major proteins: troponin I, troponin T, and troponin C. Troponin C is the calcium binding protein. The C indicates that, and when calcium binds, it releases the blockage so that myosin can directly interact with actin and force can be generated. And then the thick filament proteins, primarily myosin, and also we have titin, and then myosin binding protein C. Titin is the largest molecule. It runs through from Z line to Z line. It really kind of anchoring, holding the entire sarcomere. Major protein. If mutations in titin mostly related to dilated cardiomyopathy. So this is the standard. If you look at any biochemistry book, how muscle works, this is a kind of cross-bridge cycling. So in this one is very every beat, every contractility, you can see ATP comes in, binds to myosin, and then same time calcium binds to the troponin C. So then once that binds, the myosin is now exposed to interact with actin. As a result, the power stroke can be formed, and then the ATP at this time binds, and then the hydrolysis, and then it releases the ADP. So it's kind of circle every beat we do in the heart. So again, if there is any change in this cycle in the amount of ATP, the amount of calcium, that can alter cardiac function. So I put this one just for the students level, how this work. It's like our car engine. The sarcomeres, the muscles are very much builder in a way. Everything should be in a proper position, location, and the distance between two sarcomere in the myosin heavy chain, where they arrange myosin binding protein C. For example, every 11 myosin heavy chain has one myosin binding protein C. The distance between two myosin binding protein C is 43 nanometer. So everything is very much defined in the muscle. So you cannot just mess up that one. So again, you can see when calcium comes in, then it binds to actin, and then force can be generated. So this is a very defined structure. You cannot mess up the system. Once you started messing up, and then the disease or any changes in the function can be occurred. So here is the structure of these two major protein, myosin heavy chain and myosin binding protein C. This is the motor protein. We have myosin is always like a heterodimer. They always have in two different protein. So the head region, and then we have the proximal S2 region, and then the light myromycin region. Whereas myosin binding protein C, change it to laser. Myosin binding protein C, you can see here, it's also a very large protein, 150 kDa protein. At the N terminus, you have C0 domain, and then all the way to C10 domain. And again, there are various functionality. For example, in this slide, you can see the C terminus of myosin binding protein C interacts with the myosin heavy chain, whereas the N terminus reaches to actin as well as myosin heavy chain. So this interaction is so critical for regulating the amount of force myosin heavy chain can generate. We call it myosin binding protein C as a kind of switch based on how much inhibition or activation occurs, the force can be generated. Right now the light switch, I don't know whether you have here, you can increase or decrease the amount of power, the electric power. Based on that, you can have more brightness. Similar way, myosin binding protein C works against myosin heavy chain. The more attachment, the more binding to myosin, less force, the more free away from myosin heavy chain, we develop more force. So going back, so this is the definition of hypertrophic cardiomyopathy. So it is called disease of the sarcomere. Why? Predominantly mutations in the sarcomere genes are linked to develop the disease. That's what this has been named as the disease of the sarcomere. Again, these are the major sarcomeric proteins we have, and top two are beta myosin heavy chain and myosin binding protein C. And again, if we have the mutation in these two genes, it's almost a guarantee to develop the disease. So how the sarcomere dysfunction occurs? So here we have the normal sarcomere. Again, you can see the myosin heavy chain attachment to actin. Normal contractility, effective relaxation, they are very ordered in the sarcomere structure. What happens when we have dysfunction because of hypertrophic cardiomyopathy? You can see there are more myosin heavy chain attached to actin. That is called hypercontraction. So the more attachment, we generate more force. So again, hypercontraction because more actomyosin interactions, and then impaired relaxation because the cardiac tissue has very strong stiffness, and then we develop the fibrosis. So this is all part of the the hypertrophic cardiomyopathy disease process. As of today, again we have over 1,400 different defined mutations that are directly associated with development of the disease hypertrophic cardiomyopathy, and 11 or more genes are now known. Again, mostly are in the sarcomere, and two most common genes are the myosin heavy chain and myosin binding protein C. This is one of the paper we reported that we went to a local hospital, and they again in the States we have genetic screening is routine. When they identify a hypertrophic cardiomyopathy patient, they immediately do genetic screening. So we have a panel of Illumina DNA sequencing system. I think it's around $2,000. The cardiology refers them and they do the sequencing. And so we got all the data set of the families. I think we had 126 families, something like that in this study. Of that, we identified 18 genes and then 125 mutations in 14 genes Of that, 50% from myosin binding protein C and 24% from myosin heavy chain. Again, myosin binding protein C has more number of mutations. So, myosin binding protein C, again, interestingly, 85% of the mutations in myosin binding protein C are truncations. What does that mean? You have a stop codon in the middle of the gene, resulting that you are not producing the whole entire protein. So, only half of the protein you are producing, and that could cause the hypertrophic cardiomyopathy disease. And then 70% of the mutations are linked with non-obstructive hypertrophic cardiomyopathy phenotype. Again, there are two major potential mechanisms. One is haploinsufficiency, the second one is Poisson polypeptide. So, haploinsufficiency is nothing but you have two alleles. One is a mutant allele where you have a truncated myosin binding protein C, and the truncated myosin binding protein C never been exist in the sarcomere because it cannot bind in the sarcomere. So, it is been removed by the ubiquitination proteasomal system. So, resulting you are living with only one allele and you have normal protein. But is it enough? The protein you generate from the wild type allele, is it enough to maintain normal sarcomere function? It is not. That is called haploinsufficiency. So, again, when you undergo any workload or developing aging process, the one allele is not enough to give you 100% of the protein. So, again, that is kind of called haploinsufficiency. The second one is the Poisson polypeptide. I will talk about some of our experiments on that one. And again, you can express the mutant protein, and the mutant protein can go randomly binds in the sarcomere and then cause contractile dysfunction. So, now I am going into some background about what is myosin binding protein C and what are the phenotype characteristic. So, myosin binding protein C, we have three different paralogs. Paralogs are not isoforms. Paralogs are unique genes. The protein can come from their own gene. Isoforms are from the same gene, but altered splicing can cause the isoforms. So, here we have three different myosin binding protein C. Slow skeletal myosin binding protein C encoded in chromosome 12, fast skeletal myosin binding protein C encoded in chromosome 19, and then cardiac in chromosome 11. As you can see, they are like very close, similar domain structures. The round shape are the immunoglobulin domain, and this one is the fibronectin domain. So, cardiac has extra 100 amino acid C0 domain, and then a couple of phosphorylation sites, and then there is a unique insert within C5 domain. But mutations in skeletal myosin binding protein C can cause skeletal disease, not cardiac disease, because they do not express in the cardiomyocytes. So, why we have to study myosin binding protein C? Again, it is an important protein, it is a transfilament protein, it connects thin and thick filaments. Without the myosin binding protein C, sarcomere structures are altered and they do not be normal in shape. And you can see it is also heart break, it regulates the actomyosin interaction and the rate of force development. Again, if you look at the electron microscopic analysis, myosin binding protein C is very specifically localized within the Z zone of the sarcomere. So, again, there are 7 to 11 stripes. Every 11 myosin heavy chain has one myosin binding protein C. As I said, the distance between two myosin binding protein C is 43 nanometer. So, it is very defined structures in the sarcomere. What happens if you remove myosin binding protein C by knockout? So this is a mouse heart and wild type, 3 months, normal, whereas this is a heterozygous. One allele is completely removed, another allele we have, and here we have both alleles are gone. You can see there is a hypertrophic phenotype within 3 months if you don't have myosin binding protein C. So what happened when you knock out myosin heavy chain? It's embryonically lethal. What happened if you knock out troponin I? Embryonically lethal. But when you remove myosin binding protein C, you still form the heart function, but they develop severe phenotype, failing heart down the road. And also myosin binding protein C has more number of genetic mutations than any other genes right now. Okay, so again we talked about 2 different mechanisms or mutations. One is gain of function, another one is loss of function. So gain of function means more than normal function, loss of function is less than normal function. So pretty much these are the 2 major examples. In myosin heavy chain, predominantly most of the mutations resulting in gain of function. What does that mean? Myosin is now being hyperactive, more active in utilizing ATP and hydrolyzing ATP and resulting generating more force. In myosin binding protein C, when you have the mutation, it causes decreased force and then decreased ATPase activity and decreased interaction with other proteins. So this can be a simple example how to define these 2 different gain of or loss of function mutations. So looking into myosin binding protein C, again I said 70% of the mutations are truncations. They have in-frame insertion or deletion or splice site donor or acceptor site mutation or premature termination or insertion or deletion in random intronic region or exonic regions. And then only 30% are the missense mutation where you have the full length myosin binding protein C. So again, hypothesis both in mouse models and also in vitro studies, this can be various mechanisms can be found on the literature. For example, some mutations either in troponin or troponin T, for example, can cause high calcium sensitivity. Some mutations can cause poison polypeptide. Some genetic variants can activate more inflammation. Some mutation can cause direct energetic deficit. And then we talked about loss of function, gain of function, haploinsufficiency, low calcium sensitivity. So this all can be potential mechanisms for developing hypertrophic cardiomyopathy. So one of our graduate student, David Barfield, he studied haploinsufficiency using a mouse model where we have one allele is a wild type normal, another allele is completely mutant allele, and resulting there is no protein. So the model we have is just one allele but showing a very severe phenotype. The poison polypeptide is another one which I am going to present right now. It is kind of my PhD work, but we still continue the project. You can see when I started the project and where we stand on right now. So before I go into our research project, I wanted to make sure you have any questions so I can answer them, and before I jump into my research project. So far, do you have any questions for me? I don't know exactly the number. It definitely vary between animals, humans, so it's also the aging process. Mostly we look for compared to the control versus mutant, but again I don't know the number top of my head. Princess, do you know the number? I don't know how much micrometer. 2.5 micrometer, Z line to Z line.
I have one more question. Sir, you mentioned in your presentation that I have seen C3 and mentioned that 80% of the mutations are truncation. Is this the only protein that where this 85% the mutations are truncation? Are there any proteins reported in the literature where percentage mutation of truncation is that high?
Yeah, for example, titin, right? Titin, the giant protein, that has also a lot of truncation, but the disease is dilated cardiomyopathy, not hypertrophic cardiomyopathy.
And a following question of the same, when you say that truncation is a stop codon has been inserted, something like that. So in-frame deletion, addition is not likely to lead to the generation of stop codon. Point mutation is likely the reason to issue with this stop codon.
So in frame, so if you, for example, if you introduce one nucleotide, that changes the codon, right? You just move the codon, right? And then you can introduce stop codon. Or if you delete one nucleotide, that changes the in frame every 3 nucleotide, right? That can also introduce stop codon.
There was an image shown by you of the micrographs of the sarcomere structures. They were generated using EM.
The one picture, yeah, that's EM, electron microscope, yeah. TEM, yeah. Yeah, please. No, standard electron microscopic picture. Yeah, no. Yeah, I mean, if you look all the sarcomere literature, this is the one immunogold electron. Sorry. This is the one immunogold electron microscopic analysis where you stain the sections with antibodies and then you have gold, I think immunogold staining. Then you can identify the striations. The other one, this one is a very standard electron microscopic cross sections, and you take an image and then you will see it. You don't need to have any specialized technique for this one. Yeah. This is, it's in the standard textbook, you can find this one.
The role of calcium in this case, generally calcium functions as a second messenger.
Yeah.
So is this the way that calcium binding is playing the role with the myosin binding protein?
Yeah, so when you alter contractile function, the uptake and then release, or we call it the on rate or off rate of calcium binding, right? That can change. Resulting, you can have more calcium in the sarcomere that not been utilized or they've been more utilized. So that could cause cardiac arrhythmia, so we call it myofilament calcium sensitivity. We study how much calcium the myofilament required to generate off maximal force. 50% of the force, how much calcium required. Comparing to the control, you can see. So hypertrophic cardiomyopathy mostly hypercalcium sensitivity. We can also use ionoptic system where you can freshly isolate cardiomyocytes, and then you can also study the calcium transients by using very specific calcium labelings. You can see how fast calcium comes and then goes from the sarca, where calciums are stored within the sarcomere. Again, whatever you alter, based on the condition, that can change amount of calcium in the sarcomere.
Thank you. When you say haploinsufficiency, so does it mean that one of the SNP which is responsible for the disease manifestation is either missing or is less in frequency? What does that actually means?
Yeah, so I will go through the next set of real experiment in our lab. So you have two alleles, right? Haploinsufficiency comes when you have a truncation. You don't have the full length protein, you have half of the protein, and this half of the protein is not properly localized in the sarcomere. So they are always floating around. That can be removed by the UPS system. So you don't have this protein at all, right? So now you have only one allele, the wild type, and you generate whatever that allele gives you, mRNA and protein, right? So you don't have two alleles now, you have only one allele. During the aging process, the amount of RNA and protein from one allele is not enough to maintain normal structure. Yeah.
So this is particular about one particular gene or about any
Any
gene?
gene, if the protein is not present or not incorporated normally, and so you just have only, so for example, heterozygous knockout mice. Any gene you knock out and you have the heterozygous, that could be a haploinsufficiency.
So just a basic question. So you mentioned a table where you talked about the two proteins, myosin and the C protein, that in one protein if there is a mutation, those are mostly gain in function. Other one it is loss in function, right? So if a C protein is named like a brake for like muscle contraction, so if there is a mutation in the brake, it could be a gain in function or.
So it depends, right? So if you have a haploinsufficiency, now your myosin heavy chain has no brake, right? No control by C protein. It's kind of phosphorylation of C protein. So if you don't have C protein to control actomyosin interaction, so you have more hypercontraction. So that's a gain of function of the myosin heavy chain, but not myosin binding protein C, right? You don't have myosin binding protein C. Yeah. Okay, so the next set of talk, I'm going to give an example of a project that we do last 20 years. So that's going to be very interesting. So in myosin binding protein C, there are several founder mutations being reported in the literature. Again, I listed a number of different mutations. What is founder mutation? It is a mutation that is very specific to very specific ethnic group or a population or from a region. That can be a founder mutation. For example, the one from Germany, this 702, this one exists over 50 generation in a region. So that's been a founder mutation. So we are focusing on now 25 base pair deletion in intron 32 of myosin binding protein C, and that was my PhD discovery, and that's what I'm going to talk about right now. So the variant is 25 base pair deletion at the branch point exactly within the intron 32. That could result in skipping exon 33 or altered splicing of exon 33. So it's not like giving you a normal splicing. So resulting that you are modifying the C10 domain of the myosin binding protein C. So this region is so important for the protein to be properly localized within the sarcomere. If you modify this region, the protein cannot be present in the sarcomere. It will move away, it will be removed by the ubiquitination system. So followed by my work, PhD work, this is Dandabani, he is at now IISc Bangalore. He extended the study and then he collected samples from the rest of the world looking for this 25 base pair deletion. So from my study, we only focused on South India from Tamil Nadu and Kerala, but then this study extended and we got a paper in Nature Genetic in 2009. And you can see the yellow spot, that's a region the 25 base pair deletion is located or present. It's almost 4% of the population carry this variant. Again, if you based on today's population, we expect over 100 million people can live with this variant. And then the problem is another problem, 95% of the people with this variant are normal or incomplete penetrance. And if you have it and if you see the disease, predominantly it's a late onset. After 50 years, 60 years, you see the disease difference. So we kind of concluded in my study and his study that this can be a secondary risk factor. Again, we talked about hypertension, diabetic, etc. So if you have this variant plus all those secondary risk factors, you can develop the disease early on. But the question was like, how exactly the 25 base pair deletion caused the disease, and is it really caused the disease or not? We don't know. So that's what, when I started my own independent lab, we thought like, okay, we have to revisit this study. So in the US, South Asians, people from India, Pakistan, Bangladesh, Sri Lanka, so almost like 5.4 million people. I was at Loyola University of Chicago and I looked at the number in Chicago. It's almost 400,000 people living. So in my neighborhood, so if I say 4% of the 400,000 people, I have 17,000 people just in Chicago walking with this variant. So I don't need to come all the way here to do any genetic study. So I thought, okay. Then I had a summer student. This guy is Aravind Jagadishan. He was graduated from undergraduate and then joined my lab, and he wanted to do a research project. So I told him like, we need to go to our community, collect samples to screen for the variant. So he named the study called the Diastolic Dysfunction of South Asians, or DOSA study. As you know, DOSA is so popular. So when you put this kind of title, people get attracted, they come, they're curious, what is this? And so we can easily convince them to collect saliva samples or blood samples. So he went around, and so here are the region or cities. Wherever there is a Diwali event, Pongal event, any festival, we go there. We put the booth with the consent form, everything, and so we started collecting samples. So in the study we reported in 2018, we collected sample 2,401 samples we collected, we screened. Of that, 139 heterozygous, and then homozygous 5. So it was very clear that it follows the polymorphic variant definition. So again, you can see we have enough subject carrying the variant, and also we have a homozygous subject. So this is even more than what we reported from here. 6% of the population, this one carry the variant. So the question is, again in this study, we invited these genetic carriers to the hospital. We performed echocardiogram. At baseline echo, again 95% of the subject carrying the mutation were completely normal, which was very good news for them. And again, you are wondering why then we have this variant, what is happening? But the 5% of the subject carrying the variant show very severe phenotype, and they had hypertension, they had diabetic, and they were aged people. So we thought like maybe if you are 40 years, 45 years, you have the phenotype, but it's not enough to show by normal echo. So we then brought them back again to come to the hospital, where we performed, this is called bicycle stress echocardiography. You can see we had to have a cardiologist in the room, we had to have a sonographer, 2 assistants, and then my postdoc. So just to do one study, we had to coordinate so many things. And so this was started 2019, just 1 year before COVID. So again, we invited the 25 base pair deletion carriers, 10 subject with the genetic variant and 10 subject without kind of normal. We did whole genome sequence. We excluded any other genetic variants in this subject, and then we had only 25 base pair deletion. So you can see very nicely at the baseline there is no difference between the mutant and the wild type or non-carriers in cardiac function. But when you increase the stress, giving more stress stage, you can see there is a clear difference in their cardiac function. That tells that at subcellular level, you have the phenotype, but it's not enough to show at the organ level, bigger organ level in a normal baseline condition. You need extra stress to show that, So this was a striking study. So then we have made the mouse model just to overexpress the mutant protein. So what happened if you express the mutant protein? The answer was, again I showed this slide yesterday, the mouse with the mutant protein, they develop clear hypertrophic cardiomyopathy phenotype in 3 to 6 months of age. You can see they have the hypercontraction, the ejection fraction is increased, fractional shortening is increased, they have more thicker ventricular wall and small chamber size, etc. We had 2 papers, one is in Journal of Biological Chemistry, another one is Cardiovascular Research, talked about the pathogenicity of this variant. For example, here, so when we express the wild type protein and the mutant protein, so here we have a control. This is a cell culture experiment, uninfected and then the empty vector. So we express a normal level of these 2 proteins, right? And then when you solubilize the total protein from the cardiomyocytes, you can see the soluble fractions, we have more mutant protein, the wild type is very, very less. Again, MIG-TAC is a kind of extra 10 amino acid we introduced at the end terminus of both wild type and mutant, just to show the difference from endogenous versus exogenous. Lastly, in the myofilament fraction, so myofilament fraction is the non-soluble fraction, where all the proteins are intact, where you can see clearly that the wild type is only present, whereas the mutant protein is almost very minimum, meaning that the mutant protein is not incorporated, it's not binding in the sarcomere, it's expressed, it is there, but not in a proper position or location. So again, this could be a Poisson polypeptide effect. So when you do confocal image with this specific antibodies to see where this mutant protein localized in the sarcomere, you can see compared to uninfected control, the wild type is very normally, you can see the doublets, very normal incorporation, whereas the mutant protein is distributed, diffused within the sarcomere. So again, this could cause severe phenotype development because the muscle cannot properly function. Now we have this organoid. We have now iPSC-derived cardiomyocytes from patients or from subject carrying the variant, and we can convert them into small spherical organ-like structure. In one Petri dish, we can have 36 organoids, so you can really multi-scale, and you can do various studies like a drug screening or compound testing or phosphorylation testing. Anything you want to screen, you can use this model. You can measure the function, everything. Again, we have a paper this last year published just to establish the technique, and we can use this model to study hypertrophic cardiomyopathy. Any questions so far before I go into what we are doing right now?
Sir, I have one question. Are you saying that this 25 base pair deletion is a founder mutation in the population of South Asia?
Correct. Correct. It is.
So the question is like, can we check that if this 25 base pair deletion is evolving with the evolution of human being? Like, can we talk about the selection pressure on this particular mutation in the population? Like, if the selection pressure is, you can say, if it is helping, or if it is beneficial.
Yeah.
I know it is causing the phenotype, disease phenotype, but if it is being selected, then there is always a chance that in future we will have more burden of the disease. So can we check on those lines?
So we really look back at what time in the evolution this 25 base pair, because
So sir, sorry. So that can be checked by the allele frequency which is there in the population, whether it is already established itself in the population, or
we have established. So again, I want to go back. We determined based on the SNPs and the age, like at what
the age of the
you know, in the evolution, around 25,000 to 35,000 years ago. I think after we migrated from Africa, someone got it and then distributed. The reason why I'm saying is, from Pakistan to all the way to Kanyakumari, the distribution is the same across all the religion, all the community, all the caste system. There is no difference. It is there 4%.
Yeah.
It's not been very selective in one population or one community or one caste system.
No, but all the Pakistan and everyone, we call us as a Asian population.
Yeah.
So if the selection is happening for that, then everyone, whosoever comes in this region of the world, they are under that pressure.
Correct.
So
yeah. So the selection process, I don't know whether there is any beneficial, why we had to carry this variant this many years, if it is not beneficial, right? We don't know the answer. Again, it's kind of, you can imagine, we have to do a very systematic study to answer that.
Now, why I'm asking, now we have the 1,000 genome sequencing database.
Yes.
So if it can be exploited to answer that question.
Probably, yeah. Maybe that's a great project for a student here. What do you think?
I don't know. I just shared my view. Like,
really?
If
Yeah,
it can
no,
be.
I mean, it's a computer. I mean, they can also look all other, the UK Biobank database,
Exactly.
all other database, put together a nice study, yeah. Yeah, great suggestion, yeah.
Thank you.
Any other questions? So again, you can see I started this project 1996. So we are still going on without knowing exactly what this mutation is doing in our population. So the animal model I showed was a transgenic model. We used alpha myosin heavy chain promoter to overexpress the mutant protein, and so again we wanted to understand exactly the mechanism and how this 25 base pair deletion in intronic region, not within exon, intronic region caused the disease. So I moved a year ago to Arizona. So again you can see a lot of cactus. It's a very nice place to visit. I recommend strongly to come and visit me. So we have now two different models. So we created a humanized mouse model in which we removed the intron of mouse and we put the human intron with or without the 25 base pair deletion. So then we can see really the splicing, you know, the RNA expression, then also the phenotype presence. So you can see in the DNA sequence here, this is a splice branch point. The deletion is here in the polypyrimidine track is almost completely knocked down. So which is a critical region for splicing. So what we found, so again within, so here we have the cardiac function, and there is a clear phenotype in these mice. They develop heart failure, they show hypertrophic phenotype, and so the mutation either you overexpress or have introduced as knock-in, you can see a phenotype. So meaning that this is the genetic variant with pathogenesis. We also looked into challenging these mice to the catecholamines, stress, and see whether they show more cardiac arrhythmias compared to wild type control, and the answer is yes. We do see a lot of you know the PVCs and also the NSVT, non-sustained ventricular tachycardia. So again these mice are now been characterized by my another postdoc. We look for what exactly at the splice donor and acceptor site, how the deletion is causing the mutant protein. So in normal splicing, we have the splice donor and then splice acceptor site, and this is the region we have the mutation. And in that region, you can see there are so many transcriptional factors that are involved in like binding and then transcribing normal mRNA. The RNA binding motif proteins are key proteins. You have to have normal binding and then post-translational modifications. So again in the literature you can see various alter splicing in heart failure, in any kind of disease process. Pre-mRNA, you can have a normal splicing, and then you can see complete exon skipping, alter splicing, alter 3 prime, this is alter 5 prime, intron retention, and then mutually exclusive exon. So again, these are the various types of alter splicing. And again, we know that from the literature, if anything changes in these, and then also mouse models with alter the RNA binding proteins can also lead to develop heart failure. So we then look into you know the myosin binding protein C, again using the database, how many genetic variants been reported in the literature on the splice acceptor and splice donor side. Clearly there are plenty of mutations. So again it's an important regions to study. Here coming back to the 25 base pair deletion, this is an in vitro experiment where we used exon trapping experiment. So we cloned the region with the intron, and then exon 32, then intron 32 with or without the variant. It's a in vitro experiment using exon trapping plasmid construct system. H9C2 cells was used. We transfect And then after 48 hours, we collected mRNA and then look for the alter splicing. So clearly, the 25 base pair deletion causes alter splicing. It's guaranteed, it's there. Also in the mouse, so we go back to the knock-in mice, we also see there is a clear splicing, alter splicing present also in the mouse model carrying the 25 base pair deletion. So this is an RNA-seq. Again, you can see the splicing, all the potential splicing occurs with the 25 base pair deletion. Here we have the normal splicing. So definitely the 25 base pair deletion causes the alter splicing. And then we also looked on the level of expression of RNA binding motif proteins. There are three major different RNA binding RBM proteins, RBM 20, 24, 38, and they are upregulated. So the more RNA binding protein present, the more alter mRNA can be present. And that's what we do see in our long RNA-seq. So again, we confirm that by doing the long RNA-seq, there are increased alter splicing in these mouse models. Currently, what we are doing is now is that we have iPSCs from human subject. I showed you the organoids. We have the mouse model. And the idea is right now we are developing antisense oligos against the mutant or alter mRNA, and that can be introduced into either the cardiomyocytes or animals, and that can be used to remove the mutant mRNA as a beneficial effect. And that's kind of we are now moving forward. We have a pattern, a provisional pattern, and the full pattern is pending right now. And we have been working on this system for now. With that, I want to stop right now and then take you any questions. The last part. So again, we have the 25 base pair deletion, intronic mutation, cause alter splicing. But again, a lot of questions we are still asking, whether how these secondary risk factors like hypertension or diabetic, will they increase the RNA binding protein motif expression that could cause more alter splicing, or aging process, for example, is it aging can also increases this RNA binding motif proteins expression level, that resulting in more altered mRNA of the myosin binding protein C. So we are looking so many secondary risk factor associations. Again, we try to answer so many questions still. Again, this is Arizona. We do a lot of hiking, mountains, so much things we can do other than just work in the lab. Okay, any questions before I conclude? And then we have our next speaker is here. Okay, we coffee or break time.